Good morning, ladies and gentlemen. So, myself, Dr. P. Samuel, Assistant Professor of Biotechnology of Ayya Nadar Janaki Ammal College, Sivakasi. So, in the video tutorial, நம்ம என்ன பார்க்க போறோம் அப்படின்னா sequence databases அதை பற்றி தான் பார்க்க போறோம். ஸோ இந்த sequence databases அப்படின்னா என்ன, அதனுடைய இம்பார்ட்டன்ஸ் என்ன அப்படிங்கிறத நான் உங்களுக்கு இந்த வீடியோ tutorial மூலிமா சொல்லித்தரேன். So what ultimately we need is a well established internet connectivity and few knowledge about the websites, right? So இதில் இப்போ நம்ம ஃபஸ்ட்டு நான் என்ன பண்ணுறேன், கூகுள் க்ரோம் ப்ரௌசரை ஓப்பன் பண்ணியிருக்கேன். ஸோ இதில் என்ன பண்ணுறேன் அப்படின்னு சொல்லி பார்த்தீங்கன்னா, ஐம் டைப்பிங் ncbi.nlm.nih.gov. ஸோ இதில் பார்த்தீங்க அப்படின்னா இந்த NCBI அப்படிங்கிறது நேஷ்னல் சென்டர் ஃபார் பயோடெக்னாலஜி இன்ஃபர்மேஷன் அப்படின்னு சொல்கிறாங்க. அடுத்தது இந்த NLM அப்படின்னு சொல்லி இங்கே ஒரு வேர்டு இருக்குது. NLM ஸ்டாண்ட்ஸ் ஃபார் நேஷ்னல் லைப்ரரி ஆஃப் மெடிசன். NIH அப்படிங்கிறது நேஷ்னல் இன்ஸ்டிடியூட் ஆஃப் ஹெல்த். அண்ட் GOV, the website is owned by the government of United States, right? So this is the home page of NCBI. Very familiar database. So there are many options. So actually we used to call this database as the gateway, right? So from NCBI you can access into any areas, but what we usually prefer is protein and nucleotide sequences. So for that, right, just drag this button so that a list will open. So from the list we can choose nucleotide, right, or else we can choose protein, right, whatever we need. But let us move one by one, right? First let me choose nucleotide sequence, right? Okay, so I am looking for nucleotide sequence. Here you have to specify the name of the organism, right? For example, I'm looking for the nucleotide sequence from mus musculus, right? So from mus musculus I am looking for the nucleotide sequence of the protein called myosin. So myosin is a muscle protein of mus musculus. So for the protein I am looking for the nucleotide sequence. So after entering all these details, just click search. Okay, so these are all the result. So what I have entered is mus musculus, right? So for mus musculus specifically I am looking for the protein called myosin, right? So for myosin from mus musculus the available records are 6101. So this is the available record for my search. And for easy analysis, choose the basis between 2000 to 4000, sorry 2000 to 5000. So this is a pretty good area for simple analysis, right? Let me choose this one. So just click here. Okay, it is searching. So let me open two other tabs, right? Okay, fine, good. Let me minimize the page size, right? One more time. Okay, so the page is loading now. See, this is the GenBank page, right? This is the GenBank page, and here you could find the result for my search, right? So the organism is mus musculus and the molecule of interest is mRNA for the protein called myosin, and this is a complete coding sequence, right? So let me go down. So this is how the page starts with locus, right, definition, accession, right, version, keywords, everything. So under locus you could find the accession number, right? Accession number is an alphanumeric combination and this is unique, and this will be the token of sequence submission, and there are 6214 bases in the mRNA molecule, and the molecule is linear in type, and it was deposited in the year 1999. And here you could find the accession number. As I told you, this is the token, and this 0.1, I mean the version, refers to the number of updates. So far the sequence is updated one number of time. And keywords, myosin, right? This is an important word. And the source is mus musculus, house mouse, and this is a very important portion, organism classification. And reference, under reference you could find the authors, the title, everything, right? So the work was published in the journal Biochemistry and Biophysics Research, right? Here the abbreviation of the journal is given, and this is PubMed ID. PubMed is the literature database. Here you could find scientific literature. This is the ID of the paper, right? And when you go down, you could find features and source, and this should also be copied while writing your practical examination, right? And when you are coming down, this is the important area, right? Origin, right? So here you could find multiple rows. See, there are 6 batches per row. Each batch contains 10 nucleotide bases. So at the end of first row there are 60 nucleotides. So the second line starts with 61, third line starts with 121, right? Multiples of 6. And at the end of the page, right, there will be a double slash, right? So this indicates, or the forward slash indicates, the page comes to an end. So this is how the result page of GenBank looks like. Another database is called EMBL, European Molecular Biology Laboratory, right? So this is the right website. I will enter into this area, right? And this is the home page of EMBL. So EMBL stands for, as I told you, European Molecular Biology Laboratory, EBI, European Bioinformatics Institute. So this is the website, ebi.ac.uk. AC means academic. So this website is only for academic purpose, and it is maintained by United Kingdom, UK. So this is the home page of EMBL. See, in practical examination, examiner used to ask the students to display or to project the home page of the databases. So this is the first thing the external examiner expects from the students appearing for the bioinformatics practical examination. So students should note down the websites in their notebook, right? It is very, very important. So here you have to mention the name, right, as what we did in NCBI, right? So what I will do, I'll just copy the things from the NCBI, right? Let me copy this same organism and same protein from NCBI and copy this in the EMBL also, right? Here I can choose what I am looking for actually, right? I am looking for nucleotide sequence, right? Search, right? Here it will search and the results will be displayed. And there are 3 primary databases. They are NCBI, EMBL and DDBJ. These are very, very important, right? Every student should know all these 3 databases, all these 3 primary databases. These are all very, very important one, right? It is loading now. Okay, right? This is how the result page of EMBL looks like. See, in case of NCBI, this is the page, right? This is the fetch result page, right? For my search, the results will be displayed like this. Whereas in EMBL, the page is looking like this. So students should understand the differences, how the result page of NCBI looks like and how the result page of EMBL looks like. All these things have to be memorized by the students, right? Here I am clicking this area, right? Okay. What is ENA browser? European Nucleotide Archive. European Nucleotide Archive, right? These abbreviations are also very, very important because examiner used to ask question from these areas. What is NCBI or expand NCBI, expand EMBL, right? Expand INSDC, right? So like this, the examiners used to ask. So I don't want to waste my time, right? European Nucleotide Archive, right? So let me dismiss this one. The page is loading now, right? Fine. And the organism, musculus, accession number, right? Molecular type is mRNA. Topology is linear in nature, right? So if I want to understand more, I'll click it. Otherwise, no need to go for this one. And see, most of the online tools or most of the tools integrated in the databases will process the sequence only if it is submitted in FASTA format, right? So how can we understand that the sequence is in FASTA format? It's very simple, right? I'll navigating the page to NCBI. Here you could find the term called FASTA, right? I'll highlight it. So FASTA stands for fast alignment, right? Fast alignment, right? Here is the page. I'll click it. The page will be loaded. So till then please wait. So okay, hope the sequence is ready now, right? So in EMBL page also the FASTA format is integrated. See, this is very important one. If the sequence, if you want to know the sequence is in FASTA format, just check for the presence of special symbols, right? See, at the beginning of the sequence you could find a greater than symbol. This is very, very important. If a sequence starts with greater than symbol, it is known that the sequence is in FASTA format. So what you have to do is just copy the sequence, right? Just copy the sequence, right click, copy, open a notepad, right? Here word wrap, then save the sequence, right? So while saving the sequence, don't forget to write the name of the organism, musculus, and the protein, myosin, right? For example, in practical exam, give your roll number. For example, 20 UT 100, right? Fine. So this is how you have to save your sequence. And one more thing I want to show you. While see, even in the GenBank page, just note this greater than symbol. This greater than symbol denotes this given sequence is in FASTA format. well. And here, right, this is very important portion actually, and here you have to note down an important thing, right? This will gives you the details about the page, right? The details you are looking for, nucleotide sequences, coding sequences, so everything you can get it from this website. And one more thing I want to show you. Well, right, and fine, wait, let me display this one. Right. Fine. Wait, this is the accession number. I am copying it and I am pasting the same in, right? And another database is also available called DDBJ, right? Let me open it in the next page. Okay, and EMBL format. See, I'll demonstrate you again. Once you are in the page, once you have make sure that you are in the correct page of your of your query, and press here, right? I want to view my result page in EMBL format, right? Okay, the page is loading now, and let me make some small manipulations in the GenBank result page, right? See the difference. This is the result page of GenBank, or this is how the page looks like. This is how the result page looks like in GenBank, and this is called the EMBL result page. Here you could find the 2 letters code, right? Here you could find the 2-letter code. This is the uniqueness of EMBL website. The 2-letter code is very important. There is an expansion for all these 2-letter codes, right? I will tell you in the next module. And this is how the result page looks like, right? This is how the result page looks like in NCBI, right? And this is the result page of EMBL, right? So far I told you how to map the home page of database. Second thing, I have displayed the result page of my queries. And the third thing I would like to explain is the DDBJ, DNA Data Bank of Japan. DDBJ stands for DNA Data Bank of Japan, right? National Institute of Genetics. This is also for academic purpose, and it is owned by Japan, JP, right? So this is the area you want to go in, right? Search and analysis, right? Search and analysis. So under search and analysis, get into ARSA, right? Okay, now we are in the correct page, and I want to enter the sequence, right? Musculus myosin. Let's go for the search, and this is how the result page of DDBJ looks like. See the differences. And one thing, see, these NCBI, DDBJ and EMBL are connected to each other. They are connected to each other through a platform called INSDC. stands for International Nucleotide Sequence Database Collaboration, right? This facilitate the exchange of sequence or exchange of information between all the three websites, right? A researcher, whatever he posted or whatever he submitted in any one of the database will be shared between the other two. This sharing is due to INSDC, right? So the students should understand the home page as well as the result page of all the databases. If I want to know further information about this sequence, right, let me click at the accession number so that it will take me to the next page, right? So this is how the result page looks like. Similarities: DDBJ and NCBI are most similar to each other, whereas EMBL is little different. As I told you, the uniqueness of EMBL is 2-letter code, right? Whereas NCBI and DDBJ looking very, very similar. And hope we'll come to the end of the session. So you should, so far I have told you, or I have introduced you 3 databases: NCBI, EMBL and DDBJ. All these 3 databases are primary sequence databases, and moreover, all these databases are connected or interconnected to each other by INSDC, International Nucleotide Sequence Database Collaboration. This collaboration facilitates the exchange of sequence information between the 3 databases, right? And I hope this video tutorial will help you and it will fulfill your perception about the databases. Thanks for watching the tutorial. Stay tuned with me. Bye-bye.